So, a very good evening to one and all present. So, we'll just wait for 2-3 minutes for others to join and then we'll start the session. Okay. Meanwhile, you can just go through and revise whatever we have learned up till now because as this is the last class, here we'll be revising by taking a few questions from each and every class. And while revising the questions, we'll also revise the concepts. So we'll just have an idea of what all we have studied up till now. So this would be very much beneficial for you for your exams per se, and you will have a revision also. Okay. So meanwhile, we are waiting for others to join. The people who are already here can join for the class. Okay. So this is going to be your last class. So yesterday we took one class where we have completed the questions from week 12, and this is the class where we'll be revising everything. Okay. So we'll just wait for 1 more minute for to just see if anybody is joining. Afterwards, we'll start. Okay. So let us start the session now. So I have made it in such a way that I have taken questions from each and every week. Okay. So I have taken questions from week 1, starting from week 1 up till week 12. That is the yesterday's class. So in that way, we'll be discussing it. So the first question is choose all the correctly matched options. Okay. And your options are P. Human and arm sea limb. Q. Bird wing and bat wing. R. Rat limb and bat limb. S. Inscus and malus. T, bird wing and butterfly wing. Okay. And you have to match it against the options which are given on the right side. That is first, pharyngeal arch, homologous organs, limb bird, and analogous organs. So it is basically a question. If you remember, we have discussed this question earlier also. So this is a question about understanding the homology or the paralogous sequences. So it is a question related to that. Okay. So I'll give you 2 minutes to answer the question and then I'll switch the screen and then we'll discuss it. Although these are all the questions that we have already discussed, you have already read. So hopefully you will be able to answer it. But even if you could not, we are here to discuss the concepts also along with it. So there is not a problem. Okay. Think think logically and then try to answer. One more minute left. Okay. So let us discuss. So the correct answer for this question is P, Q, Q ka first S1 or RS S1 and T4. So let us see PQ. So P is human arm and C limb, and it goes with P to ka second, which is homologous organs. That means Q, Q2, Q4, S1. This is the correct one, and R2. Sorry. Okay. So I'm so sorry. So human arm and C limb, it matches with homologous organs. That means human arm and C limb, they are the examples of homologous organs. Then Q4, that is bird wing and bat wing, they are the examples of analogous organs. And rat limb and bat limb, they are pharyngeal arch. Likewise, because it can have more than one correct answers, R matches with 2. That is rat limb and bat limb, they are also homologous organs. And incus and malus, they are also pharyngeal arch, and bird wing and butterfly wing are also analogous organs. Now these are the examples of homologous organs and analogous organs. This question basically dealt with the examples of homologous organs and analogous organs, but we should also know what is the actual meaning of homologous organs and analogous organs, although we have already discussed, but let us quickly revise it. So Hmm. So, human arm and sea limb, they are the examples of homologous organs as discussed. Bird wing and bat wing, they are the examples of analogous organs. Rat limb and bat limb, they are the examples of limb bird. Incus and malleus are the examples of pharyngeal arch. And bird wing and butterfly wing are the examples of analogous organs. Now, what is the meaning of homologous organs? Homologous means comes from the word which is homology. Homology means they have a same origin. That means their origin is same. Okay. So homologous basically means these are structurally similar but functionally they are different. Okay. So structurally they are similar, but they have a different function, but they have evolved from a common ancestor. This is important in homology. They have evolved from a common ancestor. Their ancestry is common, their ancestry is same, but over the due course of time they have evolved in such a way that now they perform different functions. Okay, so structurally they are similar, but now with in due course of time they have evolved themselves in such a way that now they have functioned differently. So, homologous means they are structurally similar but functionally different. And analogous means structurally different but functionally similar. Just the opposite of homologous is analogous. That means they structurally they are similar, but functionally they are structurally they are different, but functionally they are similar. For example, these bird wing and bat wing. Structurally, if you compare the structure of bird wing and bat wing, they are not same. It is different, okay? But they have evolved to perform the same function, that is fly. But they have evolved themselves differently. But differently, they have evolved in such a way that now they perform the same function of fly. Then rat limb and bat limb, limb bird. That means they are derived from same embryonic origin. Okay, it means that they are derived from the same embryonic origin. Incus and malleus, they are derived from the pharyngeal arc. Nothing too much to explain over here. And bird wing and butterfly wing. So again, see, bird wing and butterfly wing, both wings are used for what? They are used for flying. So functionally they are same, but evolutionary they have a different origin. So therefore they are called as analogous organs. So always remember the difference between homologous organs and analogous organs. Homologous organs are the organs which have a common ancestry. They have evolved from a common ancestor. But because they have evolved from a common ancestor, uh, although they have evolved from a common ancestor, their functions are different. Whereas analogous organs, the organs which do not have evolved from a common ancestor but they perform similar function, they are analogous organs. So, is this question clear to everybody? I hope this question is clear. It is. So, why I have chosen this question? Because it is an important. This is something that I really hate. Really, really hate. Anyways, my job is to do my work whether people show off a class or not. Mm. Oh, okay. So I hope this question was clear to everybody. So why I have chosen this question? Because this is an important topic, okay? This homology and analogy, it is an important topic not only for, like I always say, that not only for your examination point of view, for your this examination point of view, but in general, these are important topics that you should know. So that is why I have kept these topics so that you have an idea and you can remember. If you don't if you couldn't recall, you can at least recall that what these topics means. Fine. Now let us come to the next question. The next question says organisms containing cells from two or more organisms are called dash. It's an easy one, but easy. But there can be some confusions. I'll tell you. I'll let you know what can be the confusions, because I have seen students confusing in two terminologies. I won't tell now what are the two terminologies. First, you try to solve it. I'll switch the screen. I'll then we'll see. One more minute left. Okay. Shall I switch the screen now? Hmm? Were you able to think? Well, I am switching the screen. So, organisms containing cells from two or more organisms are called as chimera. Okay. Now, here comes important thing. Chimeric organisms and mosaic. So, what is the difference between chimera and mosaic? Can anybody tell me the difference between a chimera and a mosaic? Tell me if anybody knows. See, what is a chimera? Chimera means body contains cells from two or more genetically distinct individual. Important is that they are genetically distinct. Okay. And when I talk about, yeah, so basically, these organisms contain cells that have originated from two different distinct zygotes. Okay. So there are two different zygotes, and fusion of embryos or exchange of cells between the embryos have led to the formation of a chimera. And the genetic origin for in this case is two or more different zygotes. So two or more different zygotes, they combine and contribute to one cell population. That is your chimera. Okay. And when I talk about mosaic, this is one zygote and two or more genotype. So chimera has two different zygotes. That means cells coming from two or more, it can be more than two also, two or more distinct individuals. Whereas when I talk about mosaic, it has one zygote. That means it has two or more genotypes. Okay. So I hope this is clear. So two zygotes, involvement of two zygotes, that means chimera. One zygote but two or more genotype, that is mosaic. Okay. Now next question is, which histones are not present in the nucleosome complex? So we have already studied about nucleosome complex, and nucleosome is a octamer which is made up of some kinds of histones, XYZ histones, and some histones are not a part of the nucleosome complex. So you have to tell which histones are not a part of nucleosome complex. Your options are: first 1 H2A, 2 H2B, 3 H3A, and 4 H1. 2 minutes to answer this question and then we'll discuss it. Think what can be the answer. One more minute left. Think. Huh? 30 seconds more. Done. Shall we discuss it? Okay. So the correct answers are 2, H3A and H1. Okay. Now can you tell me why H3A and H1 are the correct answers? Can you tell me that is H3A even a histone? Hmm? Is H3A even a histone? No. H3A is not a histone molecule. That is the thing. No. See, if you remember, the histone is a histone octamer. Okay. Octamer means octamer means 8. So it has H2A, H2B, H3 and H4. 2 H2A, 2 HB, H3 and H4. And H3A is not even a histone molecule. There is no histone named as H3A. So H3A becomes out of question, first of all. And H1, is H1 a histone molecule? Yes, H1 is a histone molecule, but where this histone molecule is present, this is a linker histone. H1 is known as a linker histone. So when I say it as a linker histone, it means that it links the two histone molecules. Okay, so linker histone means something which is linking. So H1 links the two histone molecules. So H1 is a linker histone. Whereas H2A and H2B, they are the core component of the nucleosome complex. H3A is not a histone molecule and H1 is a linker histone. So I hope this is clear to you that which histones are not present in the nucleosome complex. It is H3A and H1 because H3A is not a histone molecule and H1 is a linker histone which is not present in the nucleosome complex. It actually connects the two nucleosome complexes. Okay, there's a difference. It is connecting the two nucleosome complex. It is not a nucleosome complex. It connects the two nucleosome complexes. Okay, so shall we switch to the next question? Okay, fine. So the next question is, which of the following histone modifications cannot cause transcriptional activation? Transcriptional activation. Which of the following histone modifications cannot cause transcriptional activation? That you have to tell. So the options are, the options are first methylation, demethylation, acetylation, or farnesylation. These are the four options. You have to tell that which of these histone modifications cannot cause transcriptional activation. You have 2 minutes to answer the question. First of all, let me know, are you confirmed that all are actually histone modifications? There might be a reason that, possibility that not all are histone modifications. Think, can that be even a possible thing? I think so. Okay. So the correct answer is farnesylation. Now, what is farnesylation? Can anybody tell me? What is the meaning of farnesylation? So, farnesylation is basically a lipid modification. Okay, so farnesylation is not a histone modification. It is a modification, but it is present on the lipid molecules and not on the histone molecules. So that is why farnesylation becomes the incorrect option because it is not even a histone modification. But does it means that methylation, demethylation, acetylation, all these modifications causes transcriptional activation? So in general, we know that acetylation, it causes transcriptional activation, and demethylation or methylation, it causes the repression of transcription. So we can say that okay, acetylation is correct. Acetylation, what it does is that it kind of makes the histone molecules a little loose so that it gives transcription factor some place to bind, to come and bind and cause the transcription. So acetylation for sure is a histone modification that causes the transcription activation. Likewise, demethylation also because in like there's a general saying, not saying, I mean in most of the cases, methylation, it actually causes the repression of transcription. But there is a point which need to be noted, even in the case of acetylation also, for that matter, that in general, if you learn, you will be taught that acetylation causes the activation of transcription and methylation causes the repression of transcription. So acetylation and demethylation causes the transcription activation. But in reality, it depends that which lysine residue is getting modified. That actually decides whether the modification is a active modification or a repressive modification. For example, H3K4 trimethylation. It is a methylation modification, H3K4Me3. It is H, oh god, 3 K 4 M E 3. H3K4Me3. Now it is a methylation. That means it is a, I mean, in by principle, it should be repressing the transcription, but it is an active promoter mark. That means this methylation, if it is present, it will cause the activation. So what does it means? That on third histone, the fourth lysine residue, three methyl groups are added. So like this, you read it, that in the third H3 histone molecule at the fourth lysine residue, three methyl groups are added. So H3K4Me3 is a trimethylation and it is a activation mark. So it doesn't in reality, we have to see that which lysine residue is getting methylated or acetylated, and how many methyl groups or acetyl groups are getting added to the lysine residues. That actually determines whether the modification will be an active modification or a repressive modification. But in general, both the modifications in some with their own conditions can lead to the activation of transcription, and obviously demethylation to it causes. So the correct answer would be farnesylation, because farnesylation is a lipid modification and not a histone modification. Okay. Next question is, select the correct statements regarding the regulation of mRNA. First, 5' methylation of unmethylated mRNA aids in achieving translational competency. Second, poly-A tail affects mRNA stability. Third, 3' UTR are not required for mRNA stability but are required for translational regulation. Fourth, cis-acting proteins bind to splice sites and promote splicing. And fifth, masking loops the mRNA by binding to proteins bound to the 5' and 3' ends of the mRNA and prevents translation. Okay, so these are your options. Think. You have 3 minutes to answer this question. You have 2 more minutes left. 1 more minute left. Okay. Let us discuss this now. So the correct answers are 5' methylation of unmethylated mRNAs aid in achieving transcriptional competency, poly-A tail length affect mRNA stability, and masking loops the mRNA by binding to protein bound to 5' and 3' and prevents translation. So obviously, see 5' methylation of unmethylated mRNA aids in achieving translation competency. Yes, because some of the 5' methylation, for example, H3K4 trimethylation and all these things which are present at the promoter sequences, it helps in achieving the transcriptional competency. Poly-A tail affects mRNA stability very correctly, because the more the poly-A tail, more stable would be the mRNA. And in fact, the mRNA stability keeps on degrading after each cycle, because the poly-A length also gets decreasing. So poly-A tail indeed is a measure of mRNA stability. Then uh, masking loops by the masking loops the mRNA by binding to proteins bound to the 5' and 3' ends and prevents translation. Yes, so this is also correct. That, for example, masking loops the mRNA by binding protein. So basically, many kinds of proteins bind at the 5' and 3' end of the mRNA. And it is actually important and helpful for the mRNA also, because when the mRNA goes into the cytosol has a reducing environment. Okay. So in that reducing environment, to make the cytosol uh, intact form and to prevent its integrity, it is important. So that is why this statement is also correct. So three statements are correct in this question. Okay. So next question is, yeah. So basically, cis regulatory elements and trans regulatory elements. What is the difference between cis regulatory elements and trans regulatory elements? I have included it over here, so that if you have any problems, any questions, so whenever you are going through this lecture, you can always uh, read it. So I'll give you 2 minutes. Just quickly read the difference between cis and trans regulatory elements. So what you could understood, cis regulatory elements and trans regulatory elements. Hmm? Cis regulatory elements are located on the same DNA molecule as the gene they regulate. That is your cis regulatory. Whereas trans regulatory elements are located on the different DNA molecule from the which they regulate. This is one of the most important difference between a cis regulatory element and a trans regulatory element. That cis regulatory elements are present on the same DNA molecule as they regulate, whereas trans regulatory elements they are present on different DNA molecule as they regulate. Then cis regulatory elements they are typically short DNA sequences that act as binding sites for transcription factors, and these genes or proteins are often referred to as transcription factors. So, cis regulatory elements are the binding sites of transcription factors, whereas trans regulatory elements they are referred to as transcription factors. So, cis regulatory elements are DNA sequences which are located near the gene they regulate, whereas trans regulatory elements are the genes or proteins that can regulate the expression of target genes which are located on different DNA molecules. And that is why they are mostly the transcription factors. Okay. So I hope this is clear. What is this regulatory elements and trans-regulatory elements? Now, next question, very easy one. Ethyl methane sulfonate is commonly used as a dash in genetic screens. What is ethyl methane sulfonate? Tell me. Easy, very easy one. 2 minutes to answer this question. Although this is a very easy one, you should be given only 1 minute. But chalo, I'll give 2 minutes. Okay. So let us understand the question. So ethyl methane sulfonate is a mutagen. Mutagen is a chemical, any substance which causes mutation is a mutagen, and it is a chemical mutagen. So basically ethyl methane sulfonate is an alkylating agent. Okay. It gets into the DNA bases. It gets in between the DNA bases and then it causes mutation. Fine. Next question is organize the following in the correct sequence of events: competence, development, or induction and induction. You have to organize in the correct sequence of event which comes first, whether competence comes first, induction comes first, development, and what is the sequence that you have to tell. 2 minutes and then I'll switch the screen. One more minute left. Okay. So, to answer this question, first of all, we should know the meaning of each and every terminology. Okay. So, competence. What is the meaning of competence? Competence means the ability of a cell or a tissue to respond to a signal. Okay. So, competence means the ability of a cell or a tissue to respond to a signal, to any kind of signal, to respond. And induction, induction means a signal from one group of cells changes the fate of another group of cells. That means, first, the cell should have a competence, that means the ability to sense a signal and respond to it. First thing first. And then, after sensing the signal, after responding to the signal, the cell must be able to communicate this signal from to the other cells. That is induction. So, first, the cell should be able to sense and respond to a signal, that which is known as competence, and then the cell must be able to uh, communicate the signal from itself to the other cells, which is your induction. That is, a signal from one group of cells changes the fate of other group of cells. And then, after the series of induction events, ultimately what happens is the development. Development is the downstream differentiation and morphogenesis that happens after induction. Okay, so the sequence becomes: first is competence, second is induction, and third is development. Okay, so I hope this is clear to everybody. Now, choose the correct statement regarding the JAK-STAT pathway. Phosphorylated STAT binds and activates transcription factors responsible for gene expression. Activated JAK receptors activate STAT by phosphorylating tyrosine residues. Dimerized STAT enters the nucleus and regulates gene expression. Loss of function mutation in FGFR3 results in failure to activate STAT1 signaling in thanatophoric dysplasia. A gain of function mutation in the kinase domain of JAK receptors will have the same phenotype as a loss of function mutation in the DNA binding domain of STAT. Answer this question. I will give you 3 minutes because it is a big question. Okay. First of all, what you have to do is you have to understand all the options which are given. Try to understand the options, and then if you have a basic idea about the JAK-STAT signaling pathway, then it will become very easy for you to answer this question. Okay, try it. Two more minutes left. One more minute left. Okay, so let us discuss. So first of all, we should know to understand this question, we should know what is the JAK-STAT pathway. So JAK-STAT pathway is basically involved in the differentiation of blood cells, growth of limbs, activation of casein gene during milk production. So what happens first, as you can see that ligand it binds to the receptor. So this is the receptor, which this is the receptor earlier before the ligand is not bind to the receptors are separate, and after when the ligand comes it binds to the receptor and then the receptor gets dimerized. After the receptor gets dimerized and gets phosphorylated, it phosphorylates the JAK. After JAK gets phosphorylated, it phosphorylates the STAT proteins, and then the STAT it enters into the nucleus and it causes the activation or the transcription initiation. So is this clear to everybody? It's a very easy pathway. JAK-STAT pathway. So first JAK is phosphorylated and activated, and then STAT is activated and phosphorylated. I mean phosphorylated and activated, and then STAT ultimately goes to the nucleus and it causes the transcription initiation. Okay, so accordingly, let us try to see the statements. Phosphorylated STAT binds and activates the transcription factors which are responsible for gene expression. No, because phosphorylated STAT is the transcription factor which ultimately goes inside the nucleus and binds to. So this is wrong. Activated JAK receptors activate STAT by phosphorylating at the tyrosine residues. Yes, it is correct, as we have just discussed. When the JAK, once the JAK get activated, it activates the STAT. At the Y means tyrosine residues. So the tyrosine residues gets phosphorylated. So this is correct. So this is correct. Dimerized STAT enter the nucleus and regulates the gene expression. Yes, you can see that STAT it enters the nucleus in dimerized form only. These are the STAT molecules and it is entering the nucleus in the dimerized form. So yes, that is also correct. Loss of function mutation in FGFR3 results in failure to activate STAT signaling. No. This is a concept which is given in your notebook, I mean in your PPTs. A gain of function mutation in the kinase domain of JAK will have the same phenotype as the loss of function mutation in the DNA binding domain of STAT. No, because a gain of function mutation in the kinase domain of STAT will activate the transcription. I mean, it will increase the transcription. And a loss of function mutation in the DNA binding domain of STAT, then what happens? If STAT is not able to bind to the DNA, will there be any transcription? No. So they will have actually the opposing effects and not the same effect. So this is also incorrect. So the two correct options are activated JAK receptors activate STAT by phosphorylating the tyrosine residues, and dimerized STAT enters the nucleus and regulates the gene expression. Okay, just a minute. Then the site of sperm entry marks the dash end of the embryonic axis. Which end of the embryonic axis is marked by the site of sperm entry? Easy one, not very difficult to answer. And there is not much explanation also that has to be given in this question. The site of sperm entry marks the dash end of embryonic axis. Which end of embryonic axis is marked by the site of sperm entry? Tell me. Easy. It should be easy peasy. It is the posterior end. So the posterior end of the sperm, posterior end of the embryonic axis marks the site of sperm entry. Which of the following is not a gap gene? This is an important one. I mean, something that this concept is important actually. The question, all the questions are important, but this concept is very important. Okay. So you have to be very, I mean, you have to understand this question completely in order to answer. So the question says, which of the following is not a gap gene? Okay. Options are empty spiracles, orthodentical, cruple, and bicoid. Okay. So you have to tell me which obviously the correct answer, that which is not a gap gene. And also you have to tell me that why it is not a gap gene. Whatever gene that you select, first you will say that it is not a gap gene, and why it is not a gap gene. That also you have. Okay. So you have 2 minutes to answer this question. Starting from now. Answer this. Think and try to answer the question. One more minute left. 30 seconds more. Okay. So let us answer this question. The correct answer is bicoid. So why bicoid is the correct answer? Because bicoid is not a gap gene, but bicoid is a maternal effect gene. Okay. So just give me a minute. Let's explain it. Hmm. Okay. So bicoid is basically a maternal effect gene and it is not a gap gene. So what is a gap gene and what is a maternal gene? Can anybody tell me the difference between a gap gene and a maternal effect gene? Hmm? So basically gap genes are the zygotic segmentation genes. Okay. Zygotic segmentation genes, whereas maternal effect genes are transcribed in the mother ovary before fertilization. So your maternal effect genes are present before the fertilization, which are transcribed in the mother's ovary. For example, bicoid, nanos, dorsal, and caudal. Whereas gap genes, these are the zygotic segmentation genes. For example, crupple, hunchback, giant, empty spiracles, orthodentical, etc. Okay. Next question is green fluorescent protein is expressed under auxin inducible promoter in cultured plant cells. Under which of the following conditions will GFP not be expressed upon auxin treatment? Loss of function mutation in AUX-IAA only, loss of function mutation in ARF only, loss of function mutation in 26S proteasome, administration of anti-ubiquitin antibodies, loss of function mutation in the SCF. So, what is the correct answer for this question? The correct answers are loss of function mutation in ARF only, loss of function mutation in the 26S proteasome, administration of anti-ubiquitin antibodies, and loss of function mutation in the SCF. So, the concept behind this is that the auxin is present, it causes ubiquitation and degradation of the repressor molecules, and ARF is the auxin response element. So basically what happens when auxin is present, then there would be transcription of the auxin inducible genes, okay, with the help of auxin response factor, which is known as ARF. And auxin or auxin repressors, they have the negative effect, that is they block the transcription. Okay, fine. Next question is a plant hormone dash is important for maintaining dormancy, whereas the plant hormone dash is required to break the dormancy. Very easy question. Which hormone is required for maintaining the dormancy and which hormone is required for breaking the dormancy, that you have to tell. Hmm. I'll give you 1 minute to answer this question. Okay. Okay. So the plant hormone which is required for maintaining the dormancy is abscisic acid, whereas the plant hormone which is required for breaking the dormancy is gibberellins. We have already discussed this. Next question is the trophoblast cells induce the uterine cells to form dash, the maternal portion of the placenta which is rich in blood vessels. Think. Okay. The correct answer is the decidua. So basically, trophoblast cells it induce uterine cell to form the decidual, which is the maternal portion of the placenta. Next is choose the correct statements regarding the mechanisms of evolutionary changes. Mutations responsible for differences between species majorly occur in protein sequences. Mutations in regulatory sequences are relatively dispensable. Mutations to create phenotypic variation occur in gene expressed during embryogenesis. And evolutionary relevant phenotypic variations among species result from mutations in genes expressed in the adult stage. Hmm. Think of this question. I'll give you 3 minutes to answer this question. 2 more minutes. Okay. So let us discuss this question. Choose the correct statements regarding mechanisms of evolutionary change. First is mutations responsible for differences between species majorly occur in the protein coding sequences. So what do you think that the mutations that happens they occur in the protein coding sequences? No, they do not occur in the protein coding sequences. See, always remember the protein coding sequence. The protein is always very well preserved. Okay, so there is not much that happens in the protein coding region or in the protein because protein part is mostly preserved. Whatever changes happen, most of the changes it occurs in the DNA sequences. Okay, so this is an incorrect statement. First one is an incorrect statement. Mutations in the regulatory sequences are relatively dispensable. Yes, because see, regulatory sequences are the ones which are the non-coding regions, okay, which are regulating. So they what can happen if a mutation occurs in a promoter or suppose if a mutation occurs in enhancer region, it might not be able to activate as much as it normally does. But it doesn't mean that it is completely going to do it. No, because regulatory sequences are the sequences which are the non-coding sequences, and they are also very much important. So therefore, they become indispensable, not dispensable. So if there is any mutation in the regulatory sequences, then it might create problem in the functioning or what you can say, it might create problem in the major working of the transcription also. So it cannot be a dispensable part, it is an indispensable part. Okay, then mutations to create phenotypic variation occur in genes expressed during embryogenesis. Yes, this is a correct statement. So what does it says? Mutations to create phenotypic variations. Yes, so basically if you want to have any variations that happens, uh, so those variations should happen during the embryogenesis. If any mutation has to be carried in such a way that it can create a phenotypic variation, then it should happen in the embryogenesis part only. Otherwise, it won't show the phenotypic difference. Then evolutionary relevant phenotypic variations among species result from mutations in genes expressed in the adult stage. No, it has to happen in the embryogenesis state only or in the initial stages. It cannot happen in the adult stages. So this is also a wrong statement. So the correct statement is mutations to create phenotypic variations occur in genes expressed during the embryogenesis. Okay, now I just wanted uh, this question. Just try to pay a little bit more attention to this question, and I have something important to tell you about this. Okay, I'll just come in 2 minutes. Meanwhile, you just read all the options and tell me whether AAA is has a positive impact on the transcription or a negative impact, and likewise for proteases also. because I feel that this portion we haven't covered it in detail, as much detail as we should have. So that's why. Think. Okay. So basically, you need to understand here that if auxin is present, then there would be transcription of the auxin inducible genes, which is known as auxin response factors. Okay, and auxin IAA is basically a inhibitor. Okay, so which is basically a repressor. So it will not allow the transcription to happen. So if this is present, then GFP will not be expressed. So this is important. I think I just forgot to tell you. That's why I'm telling you that this is important to note that OXIA is a repressor and it will block the transcription process. So if it is there, then transcription will not happen. This is what I wanted to tell you. Okay. So with this, we have completed all the revision questions, and I hope that this session was little useful for you. Although I have observed this time that very few attendance, which is not a very good sign. But then it's up to you. Whatever you want to do, you can do. Anyways, so with this, we have completed the lectures, all the lectures, and I think now you have the examination. So all the best for your examinations. If you have any question, you can always comment in the YouTube videos, and then I'll try to solve it. And that's it. I think that's it for the class. So a very all the best to everybody, and hopefully you all will do very well in your examinations, and you will definitely get passed in your examination. So okay, just we see how much time do we have. We have kind of 4 minutes. I'll just go through the questions if there was anything which was left. I'm just going through the slides. If you have anything, any problem, you can just ask me. This question was clear. This is homologous and logous part was also clear. This chimera and mosaic concept was also clear. Then linker histone, NH3 is not a histone molecule. Farnesylation is a lipid modification, so not needed to be included over here. Then this was the question about the mRNA and the transcription and the post-transcriptional modifications that happens. Then we discussed in brief about what is cis and trans regulatory elements. Then about the mutations and mutagens. Then competence development induction. Then we studied about the JAK-STAT pathway, how the JAK-STAT pathway works, what is the ligand, and how JAK first gets phosphorylated and activated, which ultimately activates the STAT gene, and the STAT gene then gets inside the nucleus as a dimer. So this is important to note that STAT doesn't get inside the nucleus as a monomer, but as a dimer. So STAT gets phosphorylated, it forms a dimer, and then it enters the nucleus, and then it activates the transcription. It causes transcription initiation. Then the site of sperm entry, it is the posterior site. So sperm always enters at the posterior end of the embryonic axis. And then the difference between the gap gene and the maternal genes, that also we have discussed. So bicoid is an example of maternal effect gene. Then we went ahead with the plant developmental biology, where we have discussed about the auxin pathway and how auxin and ARF plays a role into it. And then we discussed about the hormones, that which hormone is required for maintaining that dormancy and which hormone is required for breaking the dormancy. So abscisic acid is required for maintaining the dormancy and gibberellins are required for breaking the dormancy. Again, the last class was mostly related to the evolutionary developmental biology, where we have discussed the various kinds of mutations and their phenotypic changes, and how these mutations are related to evolution, that how these mutations can ultimately give rise to and accumulate and can give rise to evolutionary changes with time. So with this we have completed, I think we have covered all the lectures from lecture 1 to lecture 12. So we'll end the class now.